generate action potential. This is the next PowerPoint, but it's easier for me to lecture drawing on the board. Okay, let's review the parts of the neuron. What is this one right here? Dendrites, and then the input part, right? This is your cell body, which contains the nucleus, and remember you have RE, RU, you have ribosome, because it's very busy making neurotransmitters, right? This is your axon. What's this one? The terminal end bulb that contains your, what does it contain? Neurotransmitters, right? The neurotransmitters are stored in the terminal end bulb. And I said we have two general classes of neurotransmitters. You will call them excitatory or inhibitory neurotransmitters. Okay? You have any questions so far on what I just said? Question? Okay. So, did you notice? Look at, there's always a space. Okay? Even though when you look at pictures, it looks like they're really touching, but they're not. There is a space always. Okay? That space is called what? Synapse. This is the synapse, right? Synapse. So we're gonna give this a name. This neuron, because it's before the synapse, this is called presynaptic neuron. Okay? This will be called your presynaptic neuron. And because this neuron number 2 is after this synapse, this is called postsynaptic neuron. You got that? You understand what pre and post means? Okay? Now look at this one. For this synapse, this becomes pre and this becomes post. You understand now the word pre and post? Question? Question? No? Let's, if we have our phones on, be sure our phone is on canvas, not on Facebook right now, okay? Be easier for you to understand if we listen. Okay, so do you have any question on the word pre and post? No? So you understand this, for this synapse, this is pre-post, right? But then when we go to this synapse, we change the word post. This now becomes pre. This now becomes post, right? Okay. Okay, so I want to ask you, for this synapse, how many synapses, let me ask you, how many synapses do you see here? Only one. Very good. I'm already telling you, but I bet you want to bet $100? Somebody, one of you, at least one will count. Oh, there's 1, 2, 3, 4, there's 5 here. And in fact, an actual neuron will have several here, so they're going to write 25 or however many they count of this one. No, a synapse, if you look at the definition of synapse, synapse just means junction between two cells. So let's look at what parts of the neuron is involved in a synapse. What's this part? The axon. And what's this part? The dendrite and cell body, right? So a synapse is the junction between the axon of the presynaptic neuron and the cell body dendrite of the next neuron. That's a synapse. It's very important for us to understand these terms, because when you start looking at the nervous system test, sometimes it gets confusing. So if you remember that a synapse is made up, oh, okay, one more thing. A synapse always in here. I'm going to draw a last one right here. What's this one that's striated and multinucleated? Huh? Muscle, skeletal muscle, right? So let's say this is your gastrocnemius. So look at, this is also a synapse, right? A synapse will always involve the axon. Wherever the axon communicates with another cell, that's the synapse part. So in this case, this axon or this neuron is synapsing with this one. It's communicating with the next one. So here, now I see 1, 2, 3 synapses in my picture. You got that? Okay? You understand? Everybody, you understand?
Yes.
You have any question? You sure? Yes.
From a muscle to the next one would also be presynaptic?
No, the muscle is now going to attach to bone. So done. We're done.
Question.
Synapse always involves a neuron. Yep, axon. Very good. Always axon. Okay? Okay, so now we remember that, do you remember I told you about the cell membrane being semi-permeable, remember? And I said, because it's semi-permeable, you had to know the word intracellular and extracellular, remember that? Because there's a difference in concentration of substances inside the cell versus outside. There's, varying permeability characteristics of chemicals. So you will find some substances more concentrated inside and some substances more concentrated outside. Okay? And I also want you to remember the ATPase, the active transport. Okay? So as far as all cells are concerned, all cells in general, now don't delete the word in general. All cells have a negative charge inside. All cells. Any kind of cell will always be negatively charged inside. Because there's a predominance of proteins, and lots of proteins are negatively charged. The organelles made up of negatively charged. Okay? So that's our number one generalization. All cells are negatively charged. For neurons, that negative charge is minus 70 millivolts. This is the resting charge of neurons. A resting charge, meaning it's not stimulated yet. At rest, it has a negative 70 charge. Negative 70 millivolts. Okay? So that's called resting membrane potential. Because there's a difference, right? Resting membrane potential. Okay, you got that? Now, you have any questions so far? We're good. Now we're going to add two very important electrolytes. So there's several electrolytes we're going to study for nerve physiology. The first two are sodium and potassium. Okay? Sodium, all the way to by anywhere in the body. Sodium is always abundant outside. It's always the most abundant cation outside the cell. So the most abundant extracellular cation is always sodium, period. You're going to appreciate that when we get to acid-base balance. But this is important for nerve physiology. For inside the cell, there's also a cation, most abundant, and that is potassium. We need to know these two. Potassium, abundant inside. Sodium, always abundant outside. Lots and lots of potassium inside. Okay? You got that so far? Okay. Now, this is the cell membrane, right? This is the cell membrane of, so where is potassium? Where is the negative charge? Everywhere it's negative, okay? Minus 70, everywhere, all the way, okay? Because this is the inside the cell. You got that, okay? And where is potassium abundant? All over, all inside. There's lots of potassium all inside. And then there's lots of sodium all outside. Okay? So I always want to draw or to write here so that students remember that potassium, this is also inside the cell. This is inside, right? This is outside the cell. When you eat something salty, you bloat because sodium is always abundant outside. And if you have too much, you pee it out. Okay, now, I want you to know that the cell membrane of neurons have openings. And those openings are called gates or channels. Gates or channels. Okay. So I want you to know the first gate called leakage gate. This is the first gate you're going to get to know. There's a few gates we want to know. The leakage. Leakage gates. And leakage gates are always open. By the way, what gates are we talking about? We're talking about nothing but protein channels that allow sodium and potassium. The way these electrons can move in or out is through gates. Okay? So what is the first sodium and potassium gate we're going to know? They're called leakage gates. You got that so far? So every gate we're going to talk about, for now, will be specific to sodium and potassium. They pass through gates, not just anywhere. So here, let's say maybe this one is a gate for sodium. Maybe this one is for potassium. They have their own specific gates. Okay? And know that sodium is a fast gate. Sodium is a fast gate. And potassium is a slow gate. This is for the next gates we're going to talk about, okay? I guess leave a space, okay? Because I'm sorry, this will be for the next gates. The leakage gates are always open, so there's no such thing as fast, slow for leakage gates. This will be for the next gates we're going to talk about, okay? So if the leakage gates are always open, look at this. The question is, what is the tendency? Remember diffusion? Remember that? So because they move, this is always open, right? What will be the tendency for sodium to do? To move in to out or out to in? No. If they're concentrated here, out to in. Sodium, this is sodium.
Are they going to the negative that's inside?
Yes, they're going to go there, and then potassium is going to come out here. Because of the fact that this is always open. You got that, right? It's always open. They're just free to diffuse down their concentration gradient. However, if that happened, you will be no more negative. Does it make sense, right? If this continue to move and you reach equilibrium, there'll be no more negative 70 charge. So this is the point. There is what you call the sodium-potassium pump. The sodium-potassium pump called ATPase. It's ATP driven. So this sodium-potassium pump makes sure that the negative 70 millivolt charge is maintained. What do? They keep kicking out every sodium that tries to go in, and they keep pushing back in every potassium that tries to come out. Let's review again, right? So the point is there is a minus 70 charge for all neurons, minus 70 millivolts. That's their resting charge. And I said there's two electrolytes we're studying now, sodium and potassium. Sodium is abundant outside, potassium abundant inside. But then there's gates called leakage, and I said they're always open. So the question is, if they are always open, then the tendency is for all the sodium, it will just diffuse inside. All the potassium will diffuse outside, so they will reach equilibrium, and that will ruin your resting charge. So to keep them within their compartments, you have the sodium-potassium pump. So this sodium-potassium pump, sodium-potassium AT phase, is key to maintaining that minus 70 millivolt charge and making sure that sodium is always abundant outside and potassium is always abundant inside. Question? 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 Right? Like all of us, look at, maybe there's 50 of us here. So there's a lot of us. The tendency will then, if this gate is open, you're going to diffuse out. But then there's a bodyguard there, just kicking you back in, kicking you back in. Stay in there. Okay? So those soldiers outside, they stay out there. Anyone who tries to come in, you kick them out. That's what's going on. So we keep our charge. That's how we keep minus 7. Okay? You got that? So
yes.
Understand the leakage gate?
Yes.
Always open. Now, there are two other gates. Okay? What are these gates we're talking about? They're nothing but gates for sodium and potassium. Okay? So I'm going to erase the leakage gates. And now, I'm going to add, in addition to the leakage gates are responsible for maintaining the resting membrane potential of minus 70. Okay? Now we're going to add two more gates for sodium and potassium. One is called mechanical gate, and the other one is called chemical gate. You got that? Mm-hmm. So there are other gates here. They're closed. What do you call the gate? Mechanical or chemical. And they are gates for also sodium and potassium. They're closed. What will open them is a stimulus. So, what will open a mechanical gate? Mechanical stimulus. What will open a chemical gate? A chemical stimulus. You understand now? So here you see this one right here. Those are also gates. You can open them. So an example of mechanical stimulus, touch, right? You see, look, that's mechanical, right? See, pulling your hair, mechanical. Stretching, mechanical. What's an example of chemical? Remember acetylcholine? That's a chemical, right? Cocaine, that's a chemical. Nicotine is a chemical. Serotonin, those are chemicals. Right? Okay? So, they can be open still. Let's pretend we're going to do, how about if I touch you, what gate will open? Mechanical. So now, I'm going to write touch. There was somebody that touched you. That touch will open now mechanical gates. So now there's more gates that will open. See? More gates that open. What are these gates again? They are gates for what too? Sodium and potassium. Now here is where we have to add sodium is a fast gate and potassium is a slow gate. Why do we have to add that? So this mechanical and chemical gates for sodium and potassium. When sodium opens, it opens fast like this. It's wide open already. Whereas potassium, when it opens, it's just slow poke, slow, slow, slow. Can you see the implication of that? When the sodium is fully open, that means lots of sodium rush in. But then there's only a trickle, trickle, trickle of potassium. So I'm going to take out lots of sodium. It's already in here. See? Lots of sodium rushed in, but only a little bit of potassium came out. Still lots of potassium in there. What do you think happens to the charge? From minus 70, don't you think that will change the charge? Right? Will it make it more negative or less negative?
Less.
Yes, right? Because sodium is fast, potassium is slow, right? So that means when you have a mechanical stimulus, you open the mechanical gate, sodium opened right away. Lots of sodium is able to go in there. Potassium is opening, but it's low. So even though potassium is trickling, it's just very limited trickling. So then it will become, the charge changes. So in response to any stimulus, we are depolarizing our cell. Let's review. You got the leakage gates, right? No problem with that. Now we have two gates, mechanical and chemical gates, right? And those gates can only be opened. It's the only way to open the mechanical or chemical is in the presence of a stimulus. So here we use the stimulus touch. That opens mechanical gate. Result is you open sodium, which is fast, potassium, which is slow. Which means lots of sodium rushes in, only a little bit of potassium trickles out. Which means it's no longer minus 70, it's going to change. Anytime you change, you depolarize. Anything, any change is called depolarize. Okay? So depends on how strong the stimulus is, right? It will be, I predict from minus 70, it's going to start going down, minus 69, minus 65, you see, right? Right? We're adding positive, then minus 60, you got that? I have to write it here, minus 69, then minus 65, and then slowly, slowly, it's going to go to minus 60. You got that? You understand why we're slowly going down? Until we get to minus 55 millivolts. Minus 55 is referred to as the threshold potential. Threshold. Threshold means all you need is to bring up the charge to minus 55. When you hit that, this cell is going to fire. All you need is to introduce enough sodium or stimulate enough to bring the charge inside to minus 55. And when you hit that minus 55 in this area right here, the first segment of the axon, this part right here, the first segment of the axon has a name. It's called initial segment. The initial segment or the first segment of the axon, also called the trigger zone. Trigger zone. So once we hit minus 55 here at the trigger zone, this is going to fire. The axon is going to start generating action potential. The word action potential only happens in the axon, not here. In the dendrite cell body region, you're just depolarizing until you get to threshold. Okay? So now it's just like what they say when you're pulling the trigger, right? That's why it's called the trigger zone. When you pull the trigger, when you pull it, there's no way, there's no turning back. Right? You pull it and it's gonna fire. You're gonna say, oh, sorry. You can't do that, right? Pull the trigger, it's gonna fire. That's the same thing here. Or when you plug in the outlet, right? You plug, it's gonna come out. You can't say, oh, I'm plugging, but can you wait? Don't release it. No, you can't. It's the same thing here. You got that so far? How about, can we change this one? Maybe we write anesthesia, right? You're doing the epidural or whatever, anesthesia. So now, this is a stimulus, right? And it's going to open a gate. What gate? Chemical gate. So these two gates, they're there, but they just respond to different kinds of stimulus, right? They're just sodium and potassium. Okay, so now here, when we get to the axon, something very important about the axon. The axon does not require a stimulus to be opened. It is charge driven. They also have sodium-potassium gates. They will also have leakage. The leakage gates are all over. Okay, there's leakage gates here. That's how you get the -70 and the 80 phase, the sodium 80 phase, right? But here in the axon, you do not have chemical gates. You do not have mechanical gates. What the axon has, in addition to leakage gates, are voltage gates. That's the last gate that you need to know for now. Voltage gates. One more time. The axon will have no chemical, no mechanical gates. What does the axon have? Leakage gates, so that you have the -70 here. It also starts with -70, right? So leakage gates, yep, and voltage gates. What are voltage gates? Sodium and potassium gates also. Same thing. But we call them differently because they're not dependent on stimulus. They are charge driven. What does that mean? The first gate will open here, the first sodium will open here, and that's the trigger for the next one, for the next one, for the next one, the next one. That's how electricity passes through the wires. So what's the characteristic? It's the same. Fast sodium, slow potassium. And sodium is actually going to keep rushing in until it reaches a charge of -30 millivolts. Here, it's -30. Sorry, not -30, positive 30 millivolts. Change the minus to positive 30. You don't need to reach threshold here. You already reached action potential. It's been stimulated already. So which means just sodium will just keep rushing it. But then it hits a point where when the charge inside is no longer -70 but positive 30, this is what you call the peak of action potential. So positive 30 is referred to as the peak of action potential. All you need to do is to reach that peak. That's then the signal. When the charge inside the axon reaches positive 30, that's the signal for all these open gates to start closing up. Except for the leakage, you know that leakage is also. But all the other gates that are open, they will start closing up. But what did we say about the gates? Sodium is fast and potassium is slow. So which means when they close, look at, sodium is already closed, but potassium is slowly closing. So what does that mean? When sodium is already closed, sodium can no longer go inside. But potassium is still able to go out. That's how you're going to change or bring back the charge to -70. Got that?
Mm-hmm.
So, when we hit positive 30, right, that's our peak action potential, right, positive 30. That's the signal for sodium and potassium gates to start closing, except for leakage. Leakage is always open, right? But then sodium is fast, it's closed already, but potassium is still slowly closing. What's the implication? When it's closed, no more sodium can go in, right? Because the tendency is sodium to go in. When you close it, sodium can no longer go inside, it stays outside, but potassium is a slow poke. That means potassium can still come out. And that's how you get rid of this positive 30, and you will eventually go back to minus 70. And it goes step by step by step. It's just one after the other, right? It's not all at once. So here it was positive 30, then this will close, then this will go back to minus 70. In the meantime, this was still, it's proceeding, okay? It's proceeding all the way to the synapse. Did you get that? Guys, do you see why Gatorade is such a big business? It was invented by the Florida Gators, right? The coach there in the University of Florida. They made a lot of money. But that's because of this. Okay? Okay, before I let you go, there's one last gate. So the action potential, look at this. You're going to say, whoa. Keeps moving, keeps moving, keeps moving. What's the purpose of action potential? The purpose is to release these neurotransmitters. So when you get to the synapse area, there's a different gate that's being opened here. Do you know what it is? Calcium gate. You see how important calcium is? You see? Did you see how important calcium is for muscle contraction? Now, do you see how important it is for your brain to function? So when you get to this part at the terminal end bulb, what will open here is the calcium gates. And calcium actually goes in, it's like an usher. It goes in, and calcium is the one that will bring these neurotransmitters out. So every neurotransmitter is holding the hand of calcium. Calcium will bring it out. See? So you know three gates: sodium, potassium, and calcium. See, that's why calcium is one of the key ingredients of any multivitamin. For your kids, they have to take their yogurt, drink their milk, whatever is high calcium, so they will do well in school. Okay, so review your